Okay, so let's get started. So welcome to everyone to today's Immuseum seminar. So it's really my pleasure to welcome today's speaker, also my friend and also co-worker in NCI, Dr. Jiang Peng. So just a brief introduction about Peng. So Peng actually currently is a Earl Statman tenure track investigator in NCI, NIH. He actually finished his undergraduate school in Tsinghua University, computer science. And then afterwards, he pursued his PhD thesis in Princeton, in US, and then also finished his PhD in computer science, which following with his further postdoc training in Dana-Farber and Harvard Medical School, Harvard School of Public Health, in particular working on bioinformatics and then cancer biology. So from 2019, Peng actually got a position, started his own independent research program in NCI and focused on the cancer AI research in cancer biology. So currently, Peng's lab is particularly interested in general theme of develop infrastructure which actually transfer the knowledge from big data to inform the cancer therapy decision. And his lab particular focus in how to utilize both genomics and imaging data to identify new regulators in cancer immune invasion, particularly including how to predict immune invasion regulators by leveraging the vast amount of functional genomic data sets and the spatial transcriptomics data. Also, they try to develop machine learning infrastructures for feature selection in imaging data to understand how spatial interaction among different cells, how they can determine the anti-cancer immune response. And then he actually helped to develop a platform to generate from online database, which enable different users to leverage the vast amount of the data resources in public domains, and then to find immune invasion mechanisms in their own clinical data sets. So I think Peng is really productive, both under his PhD study, also postdoc study. I have a lot of high-profile publications, and also he got numerous honors and awards, including K99 NCI grant, and also milestone abstract awards from international cytokine and interferon societies. And now I think today Peng's going to talk about data integrative frameworks to understand how cell-cell signaling in anti-cancer immune response. So without further ado, Peng, please just go ahead. We're looking forward to your talk.
Thank you so much, Chuan, for this fantastic introduction. And also, thank you so much to Chuan and Xiaolei for giving me the opportunity to share and discuss some of our recent work with the immunology community here. And as Chuan has introduced, my background is from computer science department, and people mostly know me because of work that I developed during my postdoc, which is a type for evaluating biomarker and regulators of immune checkpoint blockade response. And currently, my research group focuses on developing the big data framework for cancer immunologists. And in other words, my goal is to make sure that immunologists here can make discoveries from reusing lots of public data. And today, I would like to introduce two recent frameworks that we have developed. The first one is called Cytosig database for analyzing cytokine signaling cascade and predicting cytokine target activities using gene expression data. And the second one is the TRAS database for analyzing marker genes of T cell resilience in solid tumors and predicting anti-tumor efficacy of T cells by their gene expression profiles. Let's start introducing the first work. As many people here know that cytokine is the basic signal that the immunology system utilizes to relay message. And the misregulation of cytokine activities cause many types of human inflammatory disease. Also, the anti-tumor immune response is regulated by many cytokine signals, which guide T cells into the tumor microenvironment to exert proper functions or block their functions. Currently, our basic tool to study cytokines are ELISA or Luminex assays to measure the cytokine release. However, the cytokine release could be very transient, and cytokine level in samples is not equal to their functional activities in recipient cells. Also, cytokine functions have two vaccine properties, which are their complexity and redundancy. So to address this challenge, we have developed a computational framework to answer two questions for immunologists. Which cytokines are regulating your gene of focus? And what are the cytokine activity in your sample? The name of our framework is called Cytoseq, representing cytokine signaling analyzer. Cytoseq has two usages. In the first usage, you can use Cytoseq as a database of cytokine regulatory relationships. You can query a gene name to view the upstream cytokine regulators or downstream target genes if the query is a cytokine. In the second usage, you can predict signaling activities for 43 cytokines through an input sample's gene expression profile. To build a database, the first thing is always data collection. There is a large number of cytokine treatment dataset available in databases such as NCBI GEO and ArrayExpress. We hypothesize that the public data could serve as a knowledge base to model signaling activities in diverse biological contexts. However, the experimental design behind each published dataset is unique, requiring very labor-intensive expert interpretation and a standardization of the metadata. To overcome these hurdles, we established a framework first called the Framework for Data Curation, which couples large-scale automatic data processing with natural language processing functions to enable semi-automatic data curation. Finally, using this framework, we have labeled more than 20,000 cytokine treatment profiles with biological replicates. So you can see that there is a strong study bias in the current data set. Some cytokines, such as TGF-beta1 or interferons, have lots of data generated from previous studies. Well, most cytokines are poorly covered in published studies. Here are some example data from our curation. Each dot here represents one profile generated from some cell model with biological replicates merged. The y-axis is the treatment cytokine, and the x-axis is the target gene. Our data collection well captured known knowledge of cytokine pro-inflammatory cascade. For example, IL-12 could induce interferon gamma, and interferon gamma will induce CXL9, 10, and 11. When we focus on the regulations among cytokines, we found two major clusters. The first cluster comprises interferons as a regulator and a few pro-inflammatory signals as target. The second group comprises a few cytokines that signals through NF-kappa B pathway as regulators and a few pro-inflammatory signals distinct from those targeting group 1 as their unique downstream target. An essential question is whether our data collection represents the cytokine regulation in human physiological relevant settings, because these data are largely generated in cell models. Therefore, we analyze the correlation between cytokine and their capture target genes in human bulk tissues. For example, here you see in the TCGA cancer cohort, the x-axis is the IL-10 gene expression, and the y-axis is the IL-10 target average expression using the target capture from our cytokine collection dataset. And they are quite positively correlated for both the IL-10 ligand expression and IL-10 receptor interaction. This positive correlation is true for most datasets from the TCGA and the GTAC cohorts, covering thousands of samples from human cancer and normal tissues. Encouraged by the high quality of our data, we analyze the regulatory patterns revealed by our data collection. For example, you can see that IL-1 beta and TNF alpha tends to co-regulate many target genes. This is consistent with their known functions through NF-kappa signaling. However, we also observed that cytokines may fight against each other through antagonizing their downstream target in about 40% pairs. For example, our analysis indicated that the BMP6 may downregulate the IL-1 beta induced target genes, such as CXL8, CXL1, and CCL2. To the best of our knowledge, BMP6 is not known as an anti-inflammatory signal that antagonizes the IL-1 beta signaling. So we performed some experimental validation or flow analysis of intracellular protein level and ELISA assay of secreted level on some human epithelial cell lines revealed that the cytokine of CXL8 and CCL2 are consistently lower in cells treated with BMP plus IL-1 beta compared to IL-1 beta treatment alone, which means that our data collection indeed revealed some new roles of BMP6 as an anti-inflammatory molecule. The data collection also enabled us to predict cytokine signaling activity using an input sample's gene expression profile. We utilized a very simple linear regression model to decompose cytokine response patterns from an input expression profile. For example, upon IL-1 beta neutralizing antibody therapy among arthritis patients, the IL-1 beta activity reduction on day 3 predicted by Cytosec correlates significantly well with the patient clinical response measured on day 15. For another example, in the interferon alpha vaccine trial among systemic lupus patients, the IL-1 activity reduction predicted by Cytosec correlates significantly negatively with the clinical response measured by the IL-1 alpha neutralizing antibody titers from the peripheral blood of treated patient. These are further examples from anti-VEGF therapy studies in cancer cohort. We predicted the VEGF signaling activities using pre-treatment expression profiles of patient tumors. And you can see that the pre-treatment activity is quite predictive of anti-VEGF survival outcome in 2 different independent clinical studies for sunitinib and bevacizumab antibodies, as expected. We comprehensively collected transcriptomic profiles from anti-cytokine therapy clinical studies or mouse models if clinical data is not available. Each dot here is one data set. The Cytosec prediction generally agreed pretty well with the clinical outcome in about 85% of the tested data set. In other words, if Cytokines predict a reduction of cytokine activity using a patient sample, the patient will typically respond to the relevant anti-cytokine treatment. A surprising result is that Cytokines predict different activities for TGF-beta1 and TGF-beta3. They all share the same receptors of TGF-beta receptor 1 to 3. Initially, we are skeptical that whether these two isoforms have different activities because they share the same receptor. To validate our computational prediction, we utilize the different neutralizing antibodies that target different isoforms of TGF-beta and use these antibodies to treat the 41 tumor models and measure the mouse tumor expression profile and do the cytokine prediction using the gene expression data. You can see that the cytokine predicted TGF-beta isoform specific activity are indeed quite consistent with the isoform specificity of different neutralizing antibodies. We also evaluated the prediction performance of Cytokines using single-cell RNA-seq data. Our gold standard is the transcription factor activity predicted using the ChIP-seq dataset and my previous software Rabbit. We found that the cytokine activity predicted is typically very consistent with the predicted activity of their known downstream transcription factors. For example, interferon IL-27 activities are very consistent with the STAT1 activities predicted by ChIP-seq data. And for most cytokines and the transcription factor combinations we curated from literature, such consistent results are true across many single-cell RNA-seq datasets from diverse disease settings. This result indicates a good quality of cytokine prediction on single-cell transcriptomic data. Finally, we applied Cytokines to study molecular features of patients with severe COVID-19 symptoms compared to mild controls. We utilized single-cell RNA-seq data collected from patient lung lavage or peripheral blood samples and searched for cytokine activities that are significantly different between severe and mild patients. For example, IL-10 activity are significantly higher among macrophage from lavage sample of severe patients than mild control and healthy controls. In contrast, type 1 interferon activity are only high among CD8 T cells from patients with mild symptoms. Overall, we found a group of signature cytokines for severe patients, such as IL-10, IL-1 beta activities are high, and in contrast, BMP6 activities are low. We can construct a more detailed regulatory network using the cytokine data collected for these signature cytokines. We found that the activated cytokines tends to co-regulate a few common target genes, such as CXCL8 and CXCL1, which are all ligands of CXCR2. Moreover, these chemokines are also target genes reprised by unactivated cytokines, such as BMP6 and IL-4. Indeed, their gene expression levels are significantly higher among severe patients, as expected. Currently, there are some clinical trials of blocking CXCL8 or CXCR2 ligands as anti-cancer therapies, some of which already passed the phase 1 toxicity test. This result indicates the potential of repurposing these candidate anti-cancer drugs as COVID-19 relieving therapies. As a summary, we have developed a computational framework to query regulators or targets in cytokine regulatory cascade. The Cytokines can reliably predict cytokine signaling activities using both bulk and single-cell transcriptomic data. Also, our data collection revealed new knowledge, such as BMP6 as anti-inflammatory signal and the potential value of anti-CXCR2 in relieving severe COVID-19 symptoms. This paper is published on Nature Method last year, and welcome to visit our website to try our online framework. Next, I will introduce another framework named TRAS, which was developed based on cytosig to specifically help the field of T-cell-based immunotherapy. Here is our motivation. Cancer immunotherapies based on T-cells has led to significant success in some fractions of patients. However, most patients with solid tumors do not respond to these immunotherapies. And CAR-T therapy is only effective in blood cancer, but not solid tumors. The key reason for this gap is that solid tumors is a very hostile environment for T-cell to work. When T-cells are trying to attack tumors will induce many immunosuppressive factors to strike back to T-cells. To overcome these limitations, we aim to develop a computational model to address two questions. First, what T-cells are more resilient in the hostile solid tumor microenvironment? And second, should a patient take the financial and health risk of cellular immunotherapy? In other words, can you tell whether their T-cell product will work before therapy, or even before making the manufactured infusion product? We develop a computational framework named Tumor Resilient T-cells, or TRAS. The TRAS model can reliably predict the clinical efficacy of immunotherapies using T-cell expression profiles. The TRAS web framework, which integrated lots of public dataset, provides a platform for evaluating T-cell biomarkers of immunotherapy response and prioritizing regulators and candidate therapeutic targets of T-cell anti-tumor efficacy. Furthermore, the TRAS model revealed FIBP as a negative marker of T-cell anti-tumor efficacy and potential therapeutic target. This paper is just accepted by Nature Medicine. Let's look at the TRAS model. Previously, as I introduced, we have developed a cytosig platform which can predict cytokine signaling activities in single-cell transcriptomic data. In this example, using a single-cell RNA-seq data from a melanoma tumor, the TGF-beta signaling activities in CD8 T cells, each dot is a CD8 T cell computed by cytosig, negatively correlate with the T cell proliferation level, inferred by the expression of cell cycle and DNA replication genes. However, this negative correlation is not always true. If we split CD8 T cells by the expression of a third gene, you will observe some different associations. For example, the negative correlation between immunosuppression and T cell proliferation is evident only for T cells with low L7R expression or high FIBP expression level. This observation shows the basic idea of TRAS model, which is searching for genes whose status can modulate the correlations between immunosuppression and T cell proliferation. The TRAS model utilizes two stages to identify gene markers of CD8 T cells that are resilient in solid tumors. The first stage quantified the level of immunosuppression imposed on the CD8 T cells in a single-cell transcriptomic dataset as the signaling activities of immunosuppressive cytokines such as TGF-beta, 12, and PGE2. The second stage utilized a variable interaction test to identify gene expression marker of T cells that are resilient to these inhibitive signals. The relationship between immune suppression, T cell proliferation, and the expression status of a third gene could be tested through the interaction covariant in a linear regression. Intuitively, this T-Rise model is searching for expression markers of T cells that are still proliferative under the immune suppression in solid tumors. Still, using the example from the melanoma tumor we showed before, we can compute one T-Rise score for each gene. Here, each dot represents one gene. The x-axis shows the T-Rise score computed for human genes, and the y-axis presents the T-Rise score, and the x-axis presents the T-cell persistence score from a previous study, finding the expression signature of tumor-infiltrating lymphocyte that can persist in solid tumors and induce a complete cancer progression. As you can see, the T-Rise score are very consistent with the previous T persistence signature in this tumor example. And across 168 tumors and 19 other cancer types, you can see this consistent correlation evidenced by area under curve larger than 0.5 random expectation. We further evaluate whether the T-Rise signature can predict anti-tumor efficacy of T cells in cancer immunotherapies. Here is our prediction procedure. We first collected single-cell transcriptomic data publicly available from 168 tumors spanning 19 cancer types. For each tumor, we can compute one vector of T-Rise gene score. Then we got the median gene score across all tumors as the overall T-Rise signature. For each T cell sample's bulk gene expression profile, we compute the Pearson correlation as the prediction value. If this score is positive, we predict the T cell sample as effective. Otherwise, we predict the T cell sample as non-effective. Please note that there is no overlap between our training data and our validation data. And our validation data are composed of three categories, including bulk T cell expression profiles from pretreatment tumors for immune checkpoint inhibitors, infusion products for CAR-Ts, and also pre-manufactured samples for CAR-T or adoptive TIL transfer therapies. Here are some examples of T-Rise prediction. Each panel presents one study that generate gene expression profile of T cells from pretreatment tumors or cellular therapy infusion product. Each dot presents one patient. Please note that all of these data sets are from single cell studies, so we create an average bulk profile for each patient. The y-axis presents the T-Rise score correlation explained before. And the color indicates the actual clinical response. As you can see in all data sets, positive T-Rise score from the pretreatment sample predict the responder status significantly better than random. There are many published T cell signatures that generate much clinical interest, such as the persistence signature discussed before and the central memory signature, TSCM. Twice, the T-Rise signature achieved a higher prediction accuracy across all clinical cohorts than all previous signature compared. We also evaluated T-Rise model predictions using bulk RNA-seq profile of pre-manufactured samples during cellular immunotherapies, such as tumor before TIL expansion or peripheral lymphocyte for CAR-T production. A positive T-Rise score correlation always indicates a significantly better survival outcome after cellular immunotherapy. Also, compared to the T-cell efficacy signatures, the T-Rise signature achieved the best prediction performance. So here, the lower risk score, the better is the prediction. Together, this result suggests that the T-Rise can enable pre-screening patients even before cell infusion product manufacture. Such advantage is particularly important for cellular therapies, which is extremely expensive and potentially very toxic. Encouraged by the clinical prediction, we further evaluate whether T-RAS model can reveal some therapeutic target to enhance T-cell immunotherapies. We prioritize the genes whose expression level are consistently associated with low T-RAS scores and poor immunotherapy outcome in solid tumors. Our analysis only focuses on negative markers because therapeutic approach, whether by compound, antibodies, or genome editing, primarily inhibit target genes. Among all human genes, FIBP ranked first as a negative marker. To the best of our knowledge, very little literature exists for FIBP, except that FIBP may bind FGF1 and that FIBP can enhance chemotherapy resistance in colorectal cancer cell lines. Therefore, the FIBP score of the T-RAS score of FIBP are consistently negative across most tumors in your collection. Also, the FIBP expression level are significantly lower in T cells from anti-PD-1 responders than values from non-responders in solid tumors. Among the tumor samples obtained before expanding tumor-infiltrating lymphocyte for adoptive cell transfer, a high FIBP expression level indicate a worse overall survival outcome after adoptive transfer. Also, the high FIBP expression in bulk tumors correlates with T cell dysfunction phenotype. Since FIBP is such an outstanding negative marker in our integrated analysis, we decide to test whether it is a regulator and a potential therapeutic target. T-RAS and other signatures analyzed here are designed to reveal marker genes of T cell functions. However, gene markers reflect associations which may arise from indirect effect rather than causality. Establishing the mechanism of regulatory causality is desirable for the development of therapeutic applications. So we first utilize the in vitro co-cultures between T cell receptor T cells from human and mouse donors and cancer cell lines with the paired antigen. The readout is cancer cell growth and cytokine release. In this example, human T cells targeting NY-ESO1 antigen were incubated with A375 cells labeled with red fluorescence color. RFP knockout in T cells presented a much better killing efficacy than control AVS1 knockout T cells. We have evaluated the killing efficacy on T cells engineered from 2 different donors and found the result is consistent. The overall effect of FIBP knockout is very similar to our positive control, CBP knockout, which is a well-known gene modification in T cells to enhance the anti-tumor efficacy and are under clinical trial right now. Also, FIBP knockout significantly enhanced the release of interferon gamma and TNF-alpha cytokines in both human and mouse donor T cells. Next, we evaluate the in vivo efficacy of FIBP knockout T cells by adoptive transfer in mice and evaluate the B16 tumor growth. As we see that the tumor size in mice transferred with FIBP knockout T cells is significantly smaller than tumors in mice transferred with control ROTA26 knockout T cells. The effect of FIBP knockout is very similar to the positive control of CBP knockout. Both FIBP and CBP knockout T cells promoted a significantly longer survival duration. To find the mechanism on why FIBP knockout can enhance T cell anti-tumor efficacy, we performed RNA-seq in mouse T cells with FIBP or ROTA26 knockout. This is IPA pathway enrichment analysis on the differential expression profile between FIBP KO compared to ROTA26 knockout. Surprisingly, all top results are related to the downregulation of cholesterol metabolism. Several genes encoding enzymes of cholesterol synthesize, such as HMGCS, SQLE, are significantly downregulated. LDLR, the receptor for low-density lipoprotein, which transport fat molecules around the body, is also downregulated. In contrast, ABCA1, which pumps cholesterol out of cells, is upregulated. We validated the RNA-seq result using flow cytometry analysis on T-cell cholesterol levels measured by Philippine 3. Indeed, cholesterol levels in T-cells isolated from mouse tumors are significantly downregulated upon FIBP knockout, but not ROTA26 or CBP knockout. Also, the cholesterol levels are significantly upregulated upon FIBP overexpression. From the previous literature, cholesterol is known to inhibit effector T-cell functions. And indeed, culturing T-cells with ROTA26 knockout controls in high cholesterol medium will inhibit the T-cell anti-tumor killing cytotoxicity. However, T-cells with FIBP knockout is quite resilient to high cholesterol levels in the medium, and tumor killing efficacy are the same in normal and high cholesterol media. So the FIBP knockout in T-cells are making T-cells more resilient to high cholesterol levels, which is a common feature of solid tumor microenvironment. Tumor cells are very flexible under nutrition metabolism. They can take cholesterol as lipid as their nutrition. However, T-cells cannot take cholesterol, lipid, and nutrition, and cholesterol will inhibit their function. And FIBP is a modification to make the T-cell resilient. Besides FIBP knockout, we also evaluated statin treatment on modulating the cholesterol level in T-cells, as statin is a very common drug for lowering cholesterol levels. Unsurprisingly, we only see the cholesterol lowering effect of statin treatment in T-cells before activation. In effector T-cells, statin treatment even elevated the cholesterol content. This result suggests that statin is not an effective approach to control cholesterol levels in effector T-cells. By the way, besides our observation, a few previous publications also showed that statin may actually induce cholesterol levels in certain cell lineage. So in summary, we developed a computational model named tumor resilient T cells or Tregs for studying anti-tumor T cell efficacy. Welcome to try our Tregs web framework, which integrated lots of public dataset to help your study with these interactive modules and functions. We also have videos available on our website to demonstrate the usage of each function module. Further, the Tregs model revealed FIBP as a negative marker of T cell anti-tumor efficacy and a potential therapeutic target. Last, this work is done together through our team effort. In particular, Yu Zhang, my first student that I'm so lucky to get, is truly brilliant. She spearheaded the Tregs project during COVID lockdown time. Also, my colleagues and collaborators at NCI generously helped us by providing us equipment and mass. Especially at the very beginning, we have neither white lab nor budget to buy equipment as a computational group. And thank you very much for your attention. That's the end of my presentation.
Okay, so thanks, Peng, for the great talk. I see Chenqi has a question. Go ahead, Chenqi.
Okay, Peng, thanks so much for the great talk. So this is Chenqi from Shanghai. So currently I'm under lockdown. So this is a really great talk for the lockdown time.
Thank you.
So I'm particularly interested in the second part of your talk when you talk about using the cytokine signature to predict cancer immunotherapy efficacy. So this is really cool. So regarding the function of FIBP, though you propose that this gene actually sort of support cholesterol metabolism, in particular cholesterol biosynthesis.
Yes.
And when you knock out, you basically suppress cholesterol level and also promote in some ways the effector function. So here is actually something I really want to clarify. So when you use statin to treat your T cells, would you see sort of inhibition of effector function or sort of gain of function of the effect of the killing and also cytokine production?
Hi, Qi. So it's your question that like whether cholesterol treatment on T cells are modulating the T cell effector functions?
Not exactly the treatment of cholesterol, because when you treat cells with high cholesterol level for a long time, probably you will inhibit, because I will think if even you are meat lover, for example, so if you eat too much meat, it's also make you feel sick. Right?
Yes.
So cholesterol need to be maintained at a certain level, and a certain level of cholesterol will be beneficial. So that's what we observed.
Yes.
So the question would be, when you use statin to treat your effector T cells, do you see loss of function or gain of function phenotype?
We see loss of function. Like a statin will inhibit the T cell proliferation in both before and after activation, and statin will significantly induce the cholesterol levels in activated T cells. So the direction is extremely reversed.
So that's how I would like to make a suggestion about how you measure the cholesterol level. So Philippine is a dye that can measure total cholesterol level, but this dye is not very reliable for FACS analysis. But it's good for imaging. So if you use Philippine imaging, it can faithfully reflect the cholesterol level, but not for FACS. So for FACS, it will be better to use another probe developed by UT Southwestern investigator. So they called it ALD4 probe.
ALD4.
Yeah, so that probe is better to measure membrane cholesterol level, in particular the so-called accessible cholesterol.
I see.
Cholesterol
so like you are suggesting AOD4 as a marker for membrane cholesterol, which is probably more relevant.
Reliable than the Philippine staining for FACS. So Philippine is okay for imaging, but not for FACS.
I see.
So this is because when you use cholesterol biosynthesis inhibitor, so usually we expect to see reduce the cholesterol level, not gain the cholesterol level, right? But Philippine somehow, it's very tricky to use Philippine because you can easily overstain your sample. So, and also I would like to remind you the cholesterol level is highly dependent on the cell size and also cell proliferation status. So you need to keep yourself at the same density. If the cell density is different between sample and sample, the cholesterol level will change. So cholesterol is a very sensitive membrane lipid sort of component, because when the cell is overcrowded, cholesterol level will be downregulated.
I see.
Okay, so be kind of careful when you measure the surface cholesterol level by either Philippine or by the ALD4 probe.
I see. By the way, Chen Qi, like since my background is not from molecular biology, so I want to ask your opinion about the oxidation assay as an alternative cholesterol measurement. That is our alternative control in the paper. Does the oxidation assay as an alternative method will also suffer from the problem you have mentioned?
Sure. See, oxidation assay, this I think is to measure, let me say, it's cholesterol level versus esterified cholesterol. Okay, so I think oxidation basically you oxidize all the free cholesterol. right? So when you firstly measure, you use a biochemical assay called Amplex Red assay to measure total cholesterol level, and then you use cholesterol oxidation to remove all the accessible cholesterol, and then when you extract this part, then you get the number of esterified cholesterol. Okay, so I think oxidization assay is sort of useful to measure free cholesterol in total cell. Okay, so it's not particularly useful to measure the level of membrane cholesterol. So membrane cholesterol is actually more faithful readout for correlated with functional state.
I see.
After T cell activation, you will see upregulation of membrane cholesterol, in particular those accessible cholesterol. So when you use the default probe, you will know what I mean. And also, if you use Philippine staining to do imaging, you will see also the increase of Philippine intensity on membrane. But for total cholesterol, it's kind of tricky because this is including the storage cholesterol. Those cholesterol are esterified, so they are stored in lipid droplet. But those fraction is mostly non-functional. It's basically a reservoir nutrient. So because you are seeing a contradictory result of statin treatment on functional readout and the cholesterol level, so that's why I want to point out maybe you need a better probe.
So maybe AOD4, like we will definitely try that. Especially you mentioned the important point, the membrane cholesterol and also the cell size, right? Like what if the cell just have different size? Like so Chen Qi, indeed, like we will just try different alternative markers and more rigorously control the other potential confounding factors here.
Yeah, so my general feeling about cholesterol metabolism, this is in particular need to be precisely controlled. Too low or too high will be harmful.
Yeah, exactly.
Certain level of cholesterol is indeed beneficial. Yeah. So I'm just curious about the real mechanism of FIBP because.
So by the way, the FIBP does not abrogate the cholesterol level. So for example, if you knock out the SREBF2, maybe that will abrogate it. So FIBP only reduce the cholesterol level to a certain level, but the cholesterol is still there. It's just to make sure the cholesterol will not raise up. Like so it's more like a fun
preventing overproduction of cholesterol.
Yeah. So FIBP is only something the knockout will make sure there's no overproduction, but still you can see cholesterol levels are still there, like from this in vivo sorted.
I know. Yeah. So the curious difference is CBL-B knockout, right? As a E3 ligase, CBL-B is a well-known negative regulator, and you also see that CBL-B knockout will enhance T cell activity.
Not very significant.
Not very significant. Yeah. Okay, but I guess in other people's assays, CBL-B can be considered as a consistent negative regulator. So in this case, maybe it will be interesting to reanalyze the cholesterol level by the new probe, right? By the default probe. And to check, what's the real effect of membrane cholesterol level by this different knockout.
Yeah.
Okay, just a few suggestions.
Sure, thank you, Chen Qi.
Yeah. Anyway, beautiful study. I really enjoyed. So the paper, when will the paper be out?
So it's just accepted. We are still waiting for proof. Like I guess maybe it still take at least another 1 month. But you can already try the website. Okay. Because the goal is to build a website so that the immunologists can query your genes, your pathways, to pull out the data from previous clinical and preclinical studies, right?
Yeah. And then very impressed by the comparison between your data set with other pre-existing data set.
Yeah. Obviously. So our prediction that we found the two outstanding signatures we have to acknowledge, which is the central memory T cells and the persistent T cells, actually they are very close, just different subtle variations.
Okay.
However, like we found out the central memory T cell signature does not work very well on infusion product. On infusion product, the effector signature is actually very important. Like so, the true value is the Treg is the robustness on immune checkpoint blockade, and the infusion product are pre-manufactured product.
Yeah, just curious, how many genes you used for this Treg?
It's a genome signature. So like every gene, we give them a weight.
Oh, okay. Wow,
yeah.
Cool. Yeah, thank you.
Thank you, Chenji.
Okay, so Dan, go ahead to ask question. Dan?
Hello, Dan, are you here?
Hello, Dan, are you there?
Dan, you have muted yourself, I think.
Yeah, you have to unmute yourself. Yeah, go ahead.
Sorry about this. Hi, very nice talk. I have a few questions about your cytokine studies. So for your cytosig database, from my study, I think it's very difficult to detect cytokine expression in human cells without stimulation.
Yes.
So is that a special feature of tumor tissues that you can detect cytokine signals in the tumor tissues?
So Dan, you mentioned a very important point. The detection of cytokines is very hard, right? Like you could observe the transient secretion, but on RNA level, you can detect nothing. So the cytosig does not aim to detect the cytokine level. Instead, this one look at the downstream targeted genes induced by cytokines. So for example, if the cytokine release have a profound and persistent effect on many downstream pathways. For example, if you observe SMART4, SMART2, SMART3, they are so activated, right? This means this cell probably already see TGF-beta. So the cytosig actually use the downstream targeted genes collected from our data collection, and to look at whether there are some downstream targeted gene activation or repression signature from your input sample's expression profile. So in this way, even cytokine release could be transient, very hard to detect. But if you look at our downstream activities, then gene expression data is pretty robust on telling you what is happening or what signals they have already seen. So does this answer your question, Dan?
Yes, that's right. And then I have a question about your COVID study. You showed the significant increase of IL-10 in macrophage in severe patients, right?
Yeah.
So, as people know, the main problem for severe COVID is because of cytokine storm. And IL-10 is a very potent immune suppressive cytokine.
Yes.
And but I also saw, actually from my data, and I found that when I activate T cells, I saw IL-10 increase. So I think IL-10 provides the intrinsic negative feedback signal over there. But in your macrophage, which do produce high level of IL-10, how about IL-6 and the TNF-alpha production?
Yes. So TNF-alpha production is significant. Like although like I use a stringent cutoff here, so it's not showing up in my summary plot, but indeed it's significant. For IL-6, I need to check. I remember it's marginally significant, but not as extreme as the top guys. By the way, Dan, I want to mention that I see a few papers published recently, including one on cancer cell, showing IL-10 is actually promoting the T cell functions. Although I'm really surprised by that study, because they use pegylated IL-10 to treat a patient, and they show that patients have better immunotherapy response. So like I guess that the IL-10 role, the role of IL-10 maybe is context specific, although I don't know whether those papers are indeed reliable. And from the target analysis, I found some surprising thing that IL-10 and IL-1 beta, they tend to co-regulate some targets sometimes. which means the role of IL-10 may not be just as simple as the like anti-inflammatory cytokine. It could do something different.
So did you look into the phenotype of these IL-10 high macrophages in COVID? Are they more like M1 or more like M2?
That's actually a good question. So I did not check this specifically. The reason is that once I got this data, I see a few papers like reported IL-10 induction in COVID sample already. Then I just did not pay too much effort on subtyping the macrophage. But I think your suggestion is indeed important. What is the polarization of this macrophage, right? Yeah. Like my guess is maybe M2 because they secrete lots of IL-10, and I don't see any interferon gamma signatures, response signature from this macrophage. But this is just my rough guess. Like, but you are right. The polarization is always something we should look at if possible.
You probably can look into some other genes rather than cytokine genes for the
yes.
M1, M2.
Yes. Yeah. Especially there are a few computational tools helping you to do this polarization analysis. So this wouldn't be too hard to look at.
Okay, great. Thanks.
Thank you, Dan.
Yeah, I have a question. Yeah, this is Yichuan Xiao speaking. Yeah.
Hi.
This is a very good talk, and I have a question about the T-resilient model. And since you know the Treg cells is also very sensitive in the tumor microenvironment for the lipids.
Yes.
And the Hongbo, Dr. Hongbo also
show. Yeah.
Chongbo. So, and the Treg cell is also a kind of a resilient cell in tumor microenvironment. So I wonder whether your model can predict the Treg cell function, or did you do some by using your model to analyze the Treg cells, and also whether another question is the FIBP, right? Also have some function in Treg to modulate Treg function in tumor microenvironment.
So by the way, let me show. Should I call you Xiao? Like
Yichuan.
Yichuan. Yeah.
Yichuan. Hi, okay. Yichuan. It seems you know what we are doing right now. So like I know you mentioned the Nature paper from Shi Hongbo lab about the lipid metabolism in Treg cells, right? And the FIBP is such a potent regulator of the lipid metabolism in effector T cells, right? How about Treg cells? That's what we are doing right now. Like we got some really striking preliminary data, although this is far from being complete. But I can just talk about a little bit here. So we found a striking thing that if you knock out FIBP in Treg cells, the naive Treg cells, activated naive Treg cells will quickly die. So this is what we observe.
So FIBP is critical for the Treg survival, right?
Yeah, it's very, very important for Treg homeostasis. And this is actually consistent with Shi Hongbo's recent Nature paper, right? Like they can take lipid as their nutrition. And after FIBP knockout, SREBP transcription factors, we will become much weaker. Both the full length and the cleavage, the cleaved version. So and also in Shi Hongbo's paper, they show that this is the master, SREBP are the master regulators of lipid metabolism. So I guess you saw that since you really know what we are following up right now. In general, what we found that FIBP is a potential metabolism switch in the tumor microenvironment. It's doing something pretty bad for the effector T cells, but the Treg really need that. And potentially cancer cells also need the FIBP for the proper survival. And the fundamental thing is that effector T cells, they are very fragile. So they don't have the metabolism, the metabolic of flexibility, like Treg and the cancer cells. Like, ideally, if they are good soldiers, you usually expect them to take whatever nutrition is available for them and take this nutrition and fat. But in reality, they are not. Yeah.
Yeah, keep going. Yeah.
But then, like for us, like we will have a dilemma. Like further following up this protein will need something like a conditional knockout mice, whatever, and that is more and more like far away from like our initial plan of like establishing a computational group. But like who knows? Like if this is indeed very important, like maybe NCI or some of our collaborators will indeed push this forward.
Okay, that's cool. Okay, so my first question is that whether your Treg model have some predictive for the Treg function in the tumor microenvironment.
So we haven't tried yet, like because like initially we only focus on CD8 T cells, and so we did not try the CD4 population. The reason is that like when you analyze a single cell cohort, like it's not that easy to separate subpopulations within CD4 like T cell population. I do see people do that. Like in most of the like single cell RNA paper, you see people still separate the Treg population. But to be honest, like those separation is really based on a very stringent threshold of clustering. So like just transcriptomically, lots of time you won't be able to really see the difference. However, like I think that it's totally possible that the Treg resilience could be modeled by a similar approach. So our next step plan is to focus on, like as you suggested, focus on the CD4 population and find some more reliable computational signatures to focus on the function aspect of the Tregs, like their metabolism, or maybe find some cell-seq data set, or even do a cell-seq by ourselves by putting a FOXP3 marker, a PD-1 marker, to pull out the Treg signature. So in short, we have not done that because of the lack of separation on just transcriptomic space. But we definitely agree with you. This is a very important thing to try out. And we will definitely work on this in the next step.
Okay, thanks. I see. Thanks.
Thank you.
And we're nice work. Okay, thank you.
Thank you.
Okay, I think next question is from Xiaolei. Before Xiaolei, I want to Peng, I want to double check with you. What is the name that Chenqi just asked you about for the surface cholesterol probe? What's the name of that?
I remember it's AOP4, right?
So let me clarify. It's ALOD4.
Oh, ALOD4.
ALOD. Okay. I just asked it for students. Just ask the question. Thanks, Chenqi.
I see. Yeah, no problem.
Yeah. Xiaolei, go ahead.
I think there's one question before me.
Yeah, just I asked the question for the student. He just wants to know what's the probe's name.
No, he is asking what's the. Okay, I see.
It's you.
Okay, good.
Yeah, go ahead.
Okay, that's great. So Jiang, this is a really fantastic talk, and I really like it. And also, I think the guy like Chenqi mentioned who can provide that probe a room. He is a really nice guy. So if you need any reagent, he is very happy to supply.
Yeah, thank you.
So I think I have two questions. One is about the FIBP. So is that like its role in regulating cholesterol synthesis is related to the FGF pathway, or it's not?
Like my gut feeling, we have not tried this specifically. I don't know, because FGF 1 is the only known binding partner. But I'm not aware of any relationship between FGF1 and the cholesterol metabolism. So my guess is no, but I have no further evidence. So we do try to do some pull down of FIBP. Like after overexpression, we got enough protein for some Western blot analysis, but T cell will all die after 4 days. So, and the NCI protein pull-down core facility service asks us to provide lots of proteins. Unfortunately, on effector T cell, we cannot do that. So I even don't know what other partners FIBP is interacting with. So the mechanism part is really not very clear right now.
Okay, cool. So the other question is if you can go to the slide that introduced the cytosig.
Yeah.
Okay, so I guess for the first usage, that's pretty straightforward. For the second one, can you elaborate a little bit? So basically, if I have one cytokine and I want to know, or could I define, or is there any specific phenotype that, well, actually, I just, yes, also registered for your website.
Yeah. So like Xiaolei, maybe the best way is to do a demo, like
yeah, that would be fun.
So for example, the second one is more like you have a bunch of transcriptomic study samples. Like it could either be differential expression between 2 conditions, or maybe a bunch of tumor samples you have somehow normalized.
Okay.
Then you submit. Each row here is the name of the sample, and each column here is a cytokine that our model can predict, and this is just the target intensity. So the second one is more like a cytokine response pattern decomposition or deconvolution from your transcriptomic study data set.
I see.
Yeah.
Okay, and for the first part, it also include that what you are talking about like the infer of which cytokine based on its downstream effect, right?
Yeah, so the first one is like a single gene query. Like if you have a gene that you are interested in. So for example, let's take SOX4. Like this is the last project I participated in during my postdoc. Like for example, you are studying a gene. You want to know what cytokines are potentially upregulating or downregulating these genes. That is a function of the first module. So what we do is we just pull out from our like curated data set that are potential what cytokines are regulating these genes, and you can see that for example, you see TGF beta 1 in lots of cell models and experimental conditions. On general, TGF beta 1 is inducing the genes that you are studying, and you do see interferon gamma and a bunch of other cytokines are really down-regulating these genes. So the first one is the more for a single gene query and hypothesis generation. You can also input a cytokine that you are interested in and see what is a potential top target genes of this cytokine. But everything is a single gene query.
I see. And I see you have a column say the quality or high quality, whether true and false. Is that based on the like what is that?
So like lots of times when you get a cytokine treatment response profile from some cell culture model, it's totally likely that the downstream targeted genes induced totally make nonsense in the human physiological settings, right? Because cell culture is very different. And especially sometimes people treat cell lines with extremely high concentration of cytokines that you will never see in a real human body system, right? Like several micromolar, whatever. Like you will see nanomolar, picomolar, that will already be very high density in human settings. So what we do is we look at the Pearson correlation between the cytokine ligand receptor and the cytokine downstream targeted genes identified from each data set in TCGA and GTEx human bulk tissue data set. So like if we don't see any positive correlation, then we assume this data set is probably a low quality data set. But we could be wrong, like because ligand or receptor expression is not equal to the protein level. So, but anyhow, this is the only quality feature that filter that we can imagine. So then like you can put in true here to only look at the filtered data set to see what whether after our quality filter, you still observe the same thing.
I see. So basically the quality is assigned to a data set.
A data set,
instead of individual pieces of.
Yes, it's a data set. You can see that it's a data set. Okay. This is the quality assigned to this data set.
Okay, great. Yeah, thanks so much for this detailed explanation, and I will ask my people to look into.
Yes, absolutely. And
advertise on the platform. I guess now a lot of people are going to check on your website.
Yeah, thank you. And by the way, for all the audience here, so at NCI, like my lab is part of the cancer data science lab, and our next year budget will depends on the citation we get from our published paper. So guys, if you want more like a database like this to help your research, please cite us. And then we'll get more budget to enlarge this data set. And also like we do listen to the requests from our collaborators to see what is the most frequent need from immunologists, and then develop our targeted database for that need. So for example, the Cytokines, like lots of collaborators always ask me this question. Then I say, okay, I'm going to develop this database. And also the TRAS, lots of collaborators at NCI always from the cell therapy community always ask me similar questions. And then we develop the TRAS database. So like, please give us citations, then we'll have more budget to develop more for you guys.
Well, absolutely.
Only is that doing strong promotion for our institute and also for the platform.
Yeah.
Well, yeah, I think this is, it's extremely helpful, and we will try to best like use this database, and
yeah, thank you.
Probably contact you if we have any.
Sure, absolutely, yeah.
Okay.
Yeah.
Cool. Thank you.
Okay, Xu, go ahead.
Hey, Jiang, very nice talk.
Hi, Xu, great to see you again. Yeah.
Good to see you. Yeah, for the second part, I was could you just elaborate a little bit more on the algorithm, how you do the inference, and also taking account of the actual gene expression value, your calculation for the Treg. And the reason I'm asking is I'm curious if you can swap your T cell repression signature to entirely different signatures to look at, for example, activation of myeloid repressions instead of T cell repression.
Yes. So like, first of all, like for first question, Xu, so this is the maybe like a good illustration. Maybe let's take this famous IL-7R, right? Like so we first look at the anti-correlation between the immune suppression measured by TGF-beta signaling or any other like PGE2 signaling, for example, and the proliferation. You typically see a strong negative correlation because that is pretty much well known. And then you just look at whether some genes can modulate this correlation. For example, you see when the IL-7R is low, you do see the negative correlation. When it's high, you don't see the negative correlation anymore. So this one could actually be tested by the linear regression. You don't need to do any cutoff. You just put the suppression, computate the gene expression as a multiplication covariant in the linear regression. Then this coefficient just tells you whether you see something like this. So, and second is, Xu, like, what do you mean by myeloid suppression? Can you elaborate? Yeah.
So in this case, you're modeling if you take, this is based on single cell or block.
Yeah,
RNC.
single cell.
Okay. So in that case, if you say you focus the population towards myeloid cells other than T cells, for example, and then you can do the same thing, right? You can calculate the cell cycle scores. You can look at the repression signatures.
I see.
For example, so, by TJ beta or IL-10 or other repression signatures, and then you can do the same analysis.
I see. So Xu, are you suggesting, for example, I look at the M1 macrophage,
right?
And then to look at how long they could survive in the tumor microenvironment, and but when they, yeah, that's actually a very good idea. So for M1 macrophage, I assume there should be something similar that could kill them or at least repress them. Right?
You can probably do a spectrum signatures between M1, M2, or even the so-called myeloid suppressor cells. right?
Yes.
Because you're cell type specific, and then you can try to look for cell type specific repression or activation signals based on, you're essentially looking for the genes that help you explain the variations from this negative correlation between proliferation and repression signatures, right?
Yeah, this is actually a fantastic idea, because M1, then the signature will not be proliferation only. It will be a differentiation. You need to make sure M1 are not polarized to something different in the tumor microenvironment, right? Yeah, then the x-axis will be some polarization signal. The y-axis will become the polarization.
Right.
And then we will see that whether there's any way to somehow modulate their polarization.
So essentially, you are using two opposite biological process signature scores, looking for the additional gene that help you explain the variation of the negative correlation.
Yes, exactly. That's pretty much the essence of the T-RES model. And by the way, like Xu, are you aware of any like adoptive cell transfer therapy of M1 macrophage?
I'm not aware of any for the moment.
I see. Yeah. So at NCI, we do have a colleague called like Rosie Kaplan. She has some like I remember she published a paper on Cell last year on like some engineering some myeloid cells, but I haven't read that in detail. But like from what you said, I probably should check. Like as you know, so like if you search very hard enough with some keyword, you'll always find some publications on this.
Yeah, we can talk more about you if you're interested, thinking along the line for different cell types using this idea. It's really cool thoughts.
Yeah, absolutely. And also, if your lab is working on some myeloid cells and waiting to collaborate, sometimes we should definitely catch up how are you doing on the children's hospital right now.
Yeah, sounds good. Yeah. We'll talk offline.
Yeah, sure.
Okay. Well, I mean, it's my question. So I have some question related to the first part, the prediction based on the program, the Cytokines. But when you validate that you're using ELISA and FACS, right?
Yes.
So my concern is because the prediction basically on the transcriptomics level, but the validation is somehow different.
Yes.
So how you reconcile this?
Yeah, so Chuan, you are very sharp. That
I'm not the reviewer. I mean, don't worry.
Yeah. Then when you asked me this question, I thought you are the reviewer. But but anyhow, you know, like I can talk it openly here. You are right. Our like predictions based on transcriptomic, and why do we validate on protein level? This is something that really bothers us, and we still don't understand right now. When we validate on transcriptomic level by qPCR, the result is very unstable. Sometimes you observe a dramatic difference, but sometimes you see no difference. And the system is extremely unstable. The only way we can make the phenomenon stable is doing protein level, intracellular protein sorting or ELISA. Then we just said, okay, let's not give us a hard time, just put the protein data. And you know, it's functional more relevant.
No,
however, this time it's actually giving us a big lesson, like protein is not equal to transcriptomic level. And we still don't understand that why we cannot observe the transcriptomic level on a very stable way. This is very puzzling.
The other thing is the Cytokines, the database you're using to build the data set is based on all human data, or it's also their mouse data?
Only human data.
I see. Okay.
Yeah.
But they're also including both bulk and then single cell?
So the Cytokines is only based on bulk data when we build the database. So it's something like this, you know, like a keratinocyte, it's treated with something and something.
Sure. But if that's the case, I'm actually curious if the Cytokines the program can also somehow to extend to analyze the single cell data?
Yes. So we evaluate this. Does bulk data predict the single cell data? So we use the STAT smart protein level predicted by ChIP-seq target as a surrogate of our evaluation standard. And we see that you typically see the consistency with the stats, model, whatever labeled by previous literature. However, like maybe the IL-4 category, I would say, to be honest, is not doing a very good job. But for other, we do see on single cell level is doing a reasonable job.
I see. Okay. Now for the second part of the TRES program. So my first thing curious is now actually you actually identify this FIBP as a negative marker. But have you ever tried to use this marker to apply the others, non-solid tumor database?
Yes.
Whether this is not correlated.
Yeah. So like we do, like for example, this chain 2021 CAR T data set is on lymphoma. So like depends on whether you will treat lymphoma as a solid tumor. Like FIBP is not predictive as a single gene. Like this one does not work in liquid cancer, but the overall Treg signature is still very predictive. So you can see that if the Treg correlation is higher than 0, its survival is better.
But then I'm actually have the next question, because the initial rationale or the motivation you describe is to actually see the resilience of the T cell in a solid tumor.
Yes.
But all the prediction you have done is based on the data you collect from the solid tumor data. Actually, can you overlap these data with the other non-solid tumor data to see, for example, the FIBP is specific for the solid tumor, or it's also generalized to all the tumors?
So FIBP does not generalize to liquid cancer. It's only for solid tumor, but some genes are generalizable. So it's just that TCF1 is very robust genes, IL7R, but FIBP is not. It's really specific for solid tumor.
But somehow you still see the significance in the leukemia patient.
Yeah, because we use the genome-wide signature correlation, right? Like, So overall, like it's still predictive, although that's not our intention, but at least this still support us that this is doing something better than random. But there's indeed a difference, like we observed.
Then the second thing probably also reflect to the protein gene level things, because when you validate this FIBP, you're using the mouse tumor model. But these FIBP, is that conservative between the mouse and human so that you can use the mouse model?
I think they are reasonably conserved. Like at least their gene length is pretty similar. And the exome structure is almost the same. And we also have some like a human donor NYES01 data to support at least you.
No. I think that the cell line in vitro cytotoxicity assays is no problem. It's just I'm curious about in vivo model, because I will saw that you using the whole immune system is pretty much the different from the human, but yeah, anyway, if it's conservative, I guess it's sort of okay. But it will always better to actually somehow using the humanized animal to test to validate such kind of target, if you actually only build the program based on the human clinical data.
Yes, so that is absolutely true, Chuan. Like a humanized model will definitely be better. Like so initially when we started this, like we actually like also make sure to look at the protein alignment, and we don't see a significant difference, so we move forward.
Oh, sure. I mean, just to say that just to keep that in mind. But we can talk more. We have humanized model in the lab. We can, if you want.
I see. Yeah, indeed. Like a humanized model will definitely be much better.
Yeah.
Like when you first start here, like we don't dare to really deep into the humanized model because
I know it's a lot of investment.
It's lots of things. But you know, like probably you should just come to lab more during her visit.
You should come to lab more, not me. Okay, okay, thanks. All right, okay. I think that's quite an intense discussion. It's almost last for an hour. So thanks again for great talk, Peng. So I think everybody now in China, I'm particularly Shanghai, stay safe. I know you guys are in quarantine, so hopefully this is gonna be over soon, and then hopefully we'll be able to see each other, in person. But anyway, thank you so much, Peng, for the great talk, and then hopefully I see you all guys next time at museum seminar next week.
Yes.
Okay, thank you. Good night. Bye-bye.
Bye.
Bye. Bye.